Hello everyone and welcome to my video lecture series. Our topic for today is going to be X-linked disorders. Okay. All those who have not seen my previous video on structure of X and Y chromosome, kindly do so because this lecture is going to be a continuation of the previous video lecture. Okay. In the previous video lectures, I'll write down some of the words which we had studied. Right. Uh, First of all, we had talked about the X chromosome. Right. Then we had talked about the Y chromosome. Then we had talked about the similar regions in between the two chromosomes which are called as homologous regions, right? And we had talked about the dissimilar regions between the two chromosomes which are called as non-homologous regions, right? Then we had talked about the genes which are present in the non-homologous regions, right? In the case of X chromosome, these genes are called as the X-linked genes. So I will write down over here the genes which are present in the non-homologous region of the X chromosome are called as X-linked genes, whereas the genes which are present in the non-homologous region of the Y chromosome, they are called as Y-linked genes. Okay, and one more thing, Y-linked genes are also called as Hollandric genes. So I will write it down over here. Hollandric genes are nothing but Y-linked genes. So all these things we had studied in the previous video lecture. Now we are going to study about X-linked genes in this lecture. Okay, so let us rub everything off. Also, if you have not yet subscribed to my channel, kindly do so. If you have liked my previous videos, do put in a like. Anyway, coming back to the topic. So we are going to study about X-linked disorders now. X-linked disorders are the disorders which are caused because of X-linked genes. Alright, and X-linked disorders are present both in males and females because males have a single X chromosome and females have two X chromosomes. That is why these disorders are present in both the genders. Now, there are two common examples of X-linked disorders. First is color blindness, right? And the other example is hemophilia. There are many other examples, but right now for this video's sake, we are going to just study these two. If you understand the logic that is present in these two disorders, then it is same in all the remaining X-linked disorders. Alright. Now, first of all, let us understand how these X-linked disorders are caused. So I will write a statement over here. Just see if you can understand the meaning. These disorders, the X-linked disorders, are caused due to a recessive gene present on the X chromosome, right? So if I see that a recessive gene is present on the X chromosome, then that will lead to the disorder, whereas if a dominant gene is present on the X chromosome, then there will be no disorder and that person, either male or female, will be normal. Okay, now let us take the example of hemophilia. Okay, and let us designate the gene H. So if I write, we are taking the example of hemophilia over here, so I will write it down. Hemophilia. Hemophilia is also called as the bleeder's disease, wherein the blood does not clot. Okay, so now I have got two genes. One is capital H and the other one is small h. Now, normally when I write the gene in capital, it is considered as dominant. So I will write over here, capital H is the dominant gene, whereas small h is the recessive gene. Correct? Now, according to the rule, if a person is having hemophilia, then small h or the recessive gene will be present on the X chromosome. So now if I ask you, okay, to tell me the genotype of a male suffering from hemophilia, right? I want to know the genotype of a male who is suffering from hemophilia. Then what will it be? It will be X. See, first of all, just let us write down the chromosomes which are present in a male, the sex chromosomes. These two chromosomes will be X and Y. So I'm going to write down X over here and Y over here. This is the male. Now, this male is suffering from hemophilia. So I will write the small h letter on top of the X chromosome. And this is a male who is affected from hemophilia. Okay, suppose if I want to write down the genotype of a male who is a normal male and who is not suffering from hemophilia, then I will be writing down X capital H Y. So this is the normal male, this is the affected male, correct? Similarly, if I want to write down the genotype of a normal female, can you tell me, all those who are watching the video, can you tell me what will be the genotype of the normal female and genotype of the affected female for the same disorder? Let us write down the genotype of the female on the right hand side. So first of all, the female is having two sex chromosomes which are X and X. I will write it over here, X and X. In case of a normal female, both the genes will be capital because capital H is the normal gene. It does not cause the disorder. So this is a normal female. Just let me write down. This is the symbol for female. Similarly, this is the symbol for male. And if I want to write down the genotype of an affected female, what should I write? So on both the X chromosomes, there will be a small h. So this is the affected female. Affected means the female who is suffering from hemophilia. Is this correct? Now, if you pay attention closely, then in case of females, there is a third possibility which can occur. And that possibility is, what if one of the X chromosome has capital H and the other has a small h? So if I write down X capital H and X small h, then what is this female? Is she suffering from hemophilia or is she normal? So such females are called as carriers, and this female is called as a carrier female. A carrier female is basically a normal female. That means she will not be suffering from hemophilia. However, she will be carrying the defective gene with her. See, she is having both the genes, capital H, small h. If you remember the law of dominance, a dominant gene overpowers the expression of a recessive gene. So since capital H is present, capital H overpowers the expression of small h, and that is why the normal gene suppresses the effect of the defective gene and the female is normal. The female does not suffer from hemophilia. However, she is still carrying the defective gene and that is why she is called as carrier. So carriers do not show any symptoms of the disease. They just carry that defective gene or the recessive gene with them. They are basically normal. So I hope all these phenotypes are clear. In the case of males, a carrier cannot be present because a male can have only two possibilities, either a recessive gene on X chromosome or a dominant gene on X chromosome. Whereas in the case of females, there are three possibilities present. So now, since we have seen this, let us see some crosses. Later on, we will also be studying about color blindness. The logic is same, right? In case of color blindness, we will be using the letter C, capital C for a normal person and small c for a person having that defective gene. Anyway, now let us rub this off, okay, and let us see a few problems. Suppose if I say, I hope everybody knows what are crosses. How do we show crosses? If you don't know about this, please watch my video on Mendel's laws. I will put a link in the description below, and also you might see a pop-up on the right side of your screen and you can click on that if you don't know how crosses are performed. I think most of you must be knowing them, so you can directly continue with this video. So now if I say, let us take the example of a cross between a normal male, right? And this is with regard to hemophilia. Normal male is crossed with an affected female. Yes. What do you think? What will happen? So first of all, let us write down the genotype. The normal male can be written as X, right? Capital H, Y. Affected female, X, small h, X, small h. These are also called as parents, right? So all these rules you have to know. Then we write down the gametes, correct? In the case of male, there are two types of gametes which are produced. One of the gametes is X capital H and the other gamete is Y. In the case of female, both the gametes produced are of the same types. So I will write down over here X small h and the other one is X small h. Okay. Now you can directly show the offsprings or you can use the Punnett square method. Punnett square method is a checkerboard method which is again explained in the video lecture of Mendel's laws. So now if we want to write down the offsprings, let us use Punnett square method. Okay. So in the case of Punnett square method, what I do is I draw a checkerboard over here, right? And I write down the gametes over here. So the male gametes are X capital H and Y, and the female gametes are X small h, X small h. So the first offspring is going to be X capital H, X small h, right? This will be what? This will be a carrier female. Then the next will be X small h, Y. This will be affected male. Then the next will be again a carrier female, which will be X capital H, X small h. And the last will be X small h, Y, which will be an affected male. So in this case, if you see, we are getting four offsprings, right? Two of them are males, two of them are females. Both the males are affected and both the females are carriers. So if we want to write down over here, we can write down offsprings, right? And the offsprings will be X, sorry, X capital H, X small h, which is a carrier female, correct? Next will be again X capital H, X small h, which is a carrier female. Then we take both the males X small h, Y, which is affected male, and the next one will be again X small h, Y, which will be affected male. So I hope you understand how we did this cross. It's very simple. Okay, we found out the gametes and then we just link each gamete from both the parents and we mix and match. So let us take the example. If we take this as the first gamete, then from affected female, if we take this, then we get X capital H, X small h, which is the first one over here, right? Then now instead of X small h over here, I take this. So now the first from male and the second from female, again you get this, which is X capital H, X small h, right? Now both the combinations of X from male are done. Now we take Y and we combine it with this, so it becomes X small h, Y, which is affected male, correct? And now we take Y and combine it with the second gamete of the female, which is again X small h, Y, and that is over here. So I hope you understand that is how we do the crosses. Basically the most important part is writing down the genotype. I hope everybody knows what is the meaning of genotype. Genotype is basically the genetic constitution of that organism. We are representing the constitution of that organism with respect to genes. That is called as genotype. Okay. So this is one of the crosses, correct? Now another example of a cross with respect to hemophilia can be, now let us take the example of a normal male and a carrier female. Alright, if you have a pen or a pencil and a notebook, you can also write this down and try to do the cross without looking over here. So now we are going to cross a normal male with a carrier female, correct? So again, writing down the genotype, normal male. So first of all, let us write over here parents, right? The genotype of normal male is X capital H Y, and carrier female will be X capital H X small h. The next thing that we do is we have to write down the gametes, correct? Can you tell me what will be the gametes? The gametes will be, one will be X capital H in the case of males, and the other one will be Y. And in the case of females, it will be X capital H, and the other one will be X small h. Right? Now the next thing is you have to combine the gametes. So let us just combine. The first combination is going to be this and this. So we get X capital H X capital H. What is this? This is a normal female. So let me write down over here normal female. Right? The next combination is going to be XH and X small h. So that is going to be X capital H X small h, which is a carrier female. Right? So out of the two females, one is carrier, one is normal. Then again, now we take the Y gamete and attach it with this gamete of the female, and we get over here X capital H Y, which is a normal male. And the last one, last combination which is possible, Y, and we attach it with X small h, and we get X small h Y, which is affected male. So when a normal male is crossed with a carrier female, you get one female carrier, one female normal. So that means both the females are normal, and out of all the sons, 50% are affected, 50% are normal, right? One is affected, one is normal. So I hope you get it, right? So in this way we can take many different examples, right? You have to know how to write the genotypes. Please remember, in the case of males, only two genotypes are possible. In the case of females, there are three genotypes which are possible. Now let us take the example of the other disorder. In case of X-linked disorders, that was color blindness, right? The entire logic will remain the same. So let us write down over here color blindness. Okay. Now again, now you have to help me out with this. We are writing down all the genotypes for the male. Okay. So normal male, if I want to write down the genotype, and if I say that I have to represent the disorder with the letter C, small c, then the normal male will be X capital C Y. Please make sure when you are writing down capital and small c, because both the letters look similar, so try to make some sort of a mark on one of the letters. Otherwise what will happen is you might get confused whether you have written down a capital letter or a small letter. Okay. Affected male can be written as X small c Y. What I do is, on top of a small letter I put a dot so that I can make the differentiation between them, right? Similarly, if I want to write down the normal female, then the normal female can be written as X capital C X capital C, right? Affected female can be written down as X small c X capital C. Again I am putting a dot, right? And finally carrier female, which is the third genotype, can be written as, oh I'm sorry, I made a mistake over here. The affected female will not be small c capital C, it will be X small c X small c. All those who could point out the mistake, well done, right? And carrier female can be written as X small c, and the other one will be X capital C. Is this clear? So the logic is same. The recessive gene will be written in small, dominant gene will be written in capital. Let us take one example, okay, and with that we will end the lecture because the logic is going to be the same. and the cross will be an affected male suffering from color blindness is crossed with a normal female. So let us write the cross down over here. Affected male is crossed with a normal female. Alright. So the genotype of the affected males, we have to write down the parent, right? Affected male can be written as X small c y, and the normal female will be written as X capital C X capital C, right? Now we have to write down the gametes. Next step, right? What will be the gametes? In case of the male, there will be two gametes. One will be X small c, the other one will be Y. See, it's very simple. Gametes basically have half the number of chromosomes. A normal cell, okay, a normal somatic cell in the human body has got 46 chromosomes, and the gamete has 23 chromosomes. So over here, if I am writing down two chromosomes to represent an individual, in case of gametes, I will just half it down, right? So it will become XC or Y. That is the logic behind writing down half the number of chromosomes as gametes, right? So two gametes, X small c or Y, and in case of female, the gametes will be again both will be same, X capital C, the other one will be X capital C. Now the next part is offsprings. Okay. First combination is we take this and this, right? So we get the genotype X small c X capital C, which is what? Which is a carrier female. So this female is carrying the defective gene for color blindness. However, she is not suffering from it. The next combination will be this and this. So again, it will be X small c X capital C, again the same result, right? Again the result will be carrier female. So let us write it down over here, carrier female. Now Y over here and X over here, so it will be X capital C Y. So that is a, I'm sorry, that is a normal male. And the last is Y and X over here, so that is again a normal male. So if you look at this cross, did you observe something? That although the father was affected, none of the sons, okay, none of the sons have the disorder, and the father passes the defective gene to all the daughters. See, the father has only one X chromosome, right? And all the daughters are going to receive that chromosome. The sons are not going to receive the X chromosome from the father. Please remember this. All the sons receive the X chromosome from their mother and the Y chromosome from their father. So if there is a scenario wherein the father is affected and the mother is normal, then all the sons will be normal because they are receiving the Y chromosome from the father, and Y is not at all related with X-linked disorders. And if the mother is normal, then the X chromosome will also be normal. So in this case, if you see, the father who is affected is passing on the defective gene to the daughters, right? And if one of these daughters marries a normal person, a normal male, then out of the two sons, one of the son will be normal, the other will be affected. So again, the daughter is passing on the gene to the son. Now let us just continue this cross. So see, now let us, I don't know if all of you are understanding what I'm trying to say. So I'll just scroll this downwards. Now what we will do is, now just take the example of both these daughters are carriers. Let us cross one of these daughters with a normal male. So the cross will become X small c, X capital C, and we'll cross it with X capital C Y, right? This is the carrier female and this is the normal male. Now in this case, if you see, I'm not writing the gametes and everything. The first possibility will be this and this, right? So that is going to be what? That will be again carrier female, right? The other possibility will be this and this. This is going to be what? This is going to be a colorblind male. So this is affected. affected male. Then the next thing, sorry, affected male. The next thing is going to be X capital C, X capital C, which is again a normal female, and the last possibility is going to be X capital C and Y, which is a normal male, right? So if you see, out of both the males, one of them, out of both the sons, one of them is affected. So again, the gene is coming from the mother. So if you see, if you take both the crosses, the father initially over here is passing on the defective gene to the daughter over here, and the daughter is passing the defective gene to the son over here. And this kind of inheritance, let me just rub everything off. This kind of an inheritance, it is called as crisscross inheritance. Crisscross inheritance. And by that, what I mean is that the father will pass on the defective gene to his daughter, and the daughter will pass on the defective gene to her son, and again the son will pass on the defective gene to his daughter, and that is so on and so forth. So it is called crisscross inheritance, right? Just let us take a look at it again. In this cross, right, this father was affected, right? He was suffering from color blindness. He passed on the defective genes not to his sons but to his daughters. You can see both these daughters are affected, right? They are not affected, they are carriers. And then if we take one of the carrier daughters and cross with a normal male, then you see over here the male is again getting affected. I forgot to put a small dot over this, and that is called as crisscross inheritance. Is this clear? So X-linked disorders are caused because of crisscross inheritance. Okay, so you can show, you can try and practice many other crosses, right? You can see the cross between an affected male and affected female, or you can see the cross between a carrier female and a normal male, right? Or both normal. So all those different combinations you can take and you will get the results. See, the disorders will change, the letters will change, but the logic will remain the same. Please remember, finally, I am repeating this yet again, that all these X-linked disorders are caused due to a recessive gene, and we have to write them down using a small letter, and the dominant gene represents the normal person. That is a person who is not having that disorder. In case of females, if both the normal and the affected genes are present, then such a female is called as a carrier. She herself doesn't suffer from the disease but can pass the gene to the next generation. So with that, we have finished this lecture. If you haven't subscribed, please do subscribe to my channel. Put in a like, do comment, share it with your friends. Thanks a lot.